Hello, good evening, student. Am I audible to you? Okay, thank you. Okay, so today we will discuss about week 12 assignment of last semester, and then we will discuss about concepts which are taught in week 12. Okay, so this is the last class, and after that, this course will be completed. Okay, we will wait for 2 more minutes. Okay, so we will start at 7:05. So other student also, if somebody want to go. Yes, ma'am. Uh, Actually, I am not aware. I am not very well aware about the examination pattern and all. But you can revise these lectures, which are uploaded by Professor Devasis, and then we will have one more summary session. Okay, so at that time you can ask if you have any doubt. Otherwise, the means all questions will be from your these lectures only. So if you go through this lecture, then you will be able to answer all the questions. Yeah, yeah, yes. You will be you will get email. Yeah, actually they asked to organize one summary session. So I have already sent a request, but date and time will be decided by NPTEL team. So it will you will be noticed. Okay, don't worry about it. Okay, it is 7:05. We should start. So it is a week 12 questions, assignments question of last semester. Okay. So first question is, which among the following is not a component of a Clark oxygen sensor? Okay. So Clark oxygen sensor, as sir already explained. Okay. This is used to sense oxygen. Okay. Why we need to measure oxygen? Because if you further analyze that you heard about this name BOD, biological oxygen demand, COD, chemical oxygen demand. Okay, so what is this means? How much oxygen is dissolved in such a system? Okay, so that you can analyze, and some amount of oxygen is necessary. Okay, for aquatic life. Okay, otherwise they will not able to breath and all things. Okay, so that's why the lower limits are essential. Okay, say, similarly in chemical oxygen demand, if some oxidation is going on, then some oxygen should be there. Okay, here also you need to measure oxygen. Okay, so there are many, and some what say biological process also, like in your blood, okay, how much oxygen is there, it should be also measured. Okay, so in many application we need to measure oxygen. Okay, in one sentence we can say that in many application we need to measure oxygen in our analyte. Okay, that's why we require oxygen sensor. Okay, so Clark designed this sensor. Okay, so this name is given to Clark. Okay, so this question is asking which among the following is not a component of Clark oxygen sensor. So before answering this question, I will just show this what is the Clark oxygen sensor. Okay, I think you are able to see this slide. If you are if slide is not changing or you have any problem, please tell me. Okay. So this is typical picture of Clark oxygen sensor. Okay. So first of all, this is your analyte. And say it is your analyte which has oxygen. Okay. And you want to measure that how much oxygen is present in your test sample, analyte or test sample, both are same. Okay. So this whole assembly whole assembly is known as a Clark oxygen sensor. So this assembly is dipped in this water, okay, or your analyte, where you need to measure oxygen percentage. Okay. So now we will discuss what is the structure of this sensor. Sorry. So if you see, first of all, this whole assembly, okay, this is the, you can say, jacket of this sensor. Then there are two electrodes. Okay, first is platinum electrode and second is this ring electrode. Okay, if you will able to see, this is whole ring. Okay, this is the complete ring. Okay, so this ring electrode is AG reference electrode. Okay, and this is the platinum electrode. Okay, these two electrodes are connected with one battery. Okay, we applied means you applied some potential from this battery. Okay, so this is negative terminal, so electron will flow in this direction. Okay, and after that electron will flow like that. Finally it will reach on this ring ring cathode, you can say, and finally it will move in this direction, and finally again it will move to positive terminal of this battery. Okay, so this is whole circuit of electron flowing. Okay, so you understood about electrode. Okay, these are two electrodes and how electrons are coming from external source and it is flowing from your cell and finally it is reaching to positive terminal. Okay, then this is this layer. If you see this layer, this is a oxygen permeable membrane. Okay, generally it is made of Teflon. Okay, what is Teflon? So it is a tetrafluorocarbon. Okay, like that. This is polymer of tetrafluoro. Okay, like that it is made. Okay, so this is Teflon. Okay, so it is it will be allowed to permit oxygen from this layer. Okay, so oxygen will only able to pass from this layer. Okay, finally your oxygen is now inside this cell. Okay, now what will happen? Okay, it will reach to platinum electrode. Okay, and at the platinum electrode it will be reduced. Okay, now let me erase. So yes. Okay, finally we reached up to here, that oxygen is come inside. Okay, from your analyte or your sample. Okay, now this oxygen will reach at platinum electrode, which is showing in red color, this one. Okay, so here oxygen in presence of, so the medium is acidic, so you will so hydrogen ion will be provided. Okay, and 4 electrons. So from where these 4 electrons are coming, see, this electron is connected with negative terminal now. Okay, So electrons are coming from your negative terminal, and finally it is providing to oxygen by the platinum electrode. Okay, this is the platinum electrode, and these two electrons are providing from the platinum electrode. And who is acquiring these two electrons? Then oxygen is acquiring the electron, and this oxygen is coming from your test sample. Okay, so finally what happened? Oxygen which is coming from your test sample in presence of proton and presence of electron is what is this? It is oxidized or reduced? Oxygen is getting reduced or oxidized? Can anybody answer? In this reaction, oxygen is reduced or oxidized? Okay, whenever hydrogen is attached to any molecule, it is reduced. Okay, so here it is reduced. Okay, so oxygen is reducing at platinum electrode. So I think first point is clear. Okay, if you see, this is oxygen. Okay, so its oxidation number is 0, but here it is H2O, so it is O minus 2. Okay, so here oxidation number is what? Minus 2. Okay, so it is 0. Oxidation number of oxygen is changing 0 to minus 2, so it is decreasing. Okay, so whenever oxidation number is decreasing, it is called as a reduction. Okay, that's why oxygen is reducing. Okay, so finally oxygen is reduced and converted in water. Then this question should be raised in your mind. Then sir, how it is sensing the oxygen? Okay, it is means it is reduction. So how it is sensing? So finally you can say that it is consuming some electron, na? 4 electron. Okay, so finally what happened? To complete this reaction, we will discuss about another electrode, which is Ag reference electrode. Okay, Ag silver reference electrode. So what will happen? Ag electrode, which is this ring electrode. Okay, this ring. Okay, it is made of Ag. Okay, so this Ag plus 4 Cl minus, which is which is come from electrolyte. Okay, it will react with silver, and silver chloride will be formed plus 4 electron. It will give 4 electron. Okay, so final, if you see here 4 electron is consumed at this electrode, but here 4 electron are produced. Okay, it is consumed here and it is produced here. So overall it is balanced. Okay, so 4 electron will be cut by 4 electrons. So overall this electron you can say neutrality is there. Okay, so finally, so what is this reaction? Ag to AgCl. Again, Ag oxidation number is what? Oxidation number of silver is 0. Any metal which has no charge means its oxidation number is 0. Whereas here oxidation number is 1 plus 1. Okay, because if you further see this salt, it will be made of Ag plus ion plus Cl minus ion. So it is positively charged. So its oxidation number is plus 1. Okay, so here to here 0 to plus 1. So oxidation number is increased. So this is the oxidation reaction. Okay, so it is oxidized finally. Okay, so okay, so at platinum electrode reduction is occurring, whereas at Ag electrode oxidation is occurring. Okay, finally this solution is KCl solution. So, okay, so if this question is arising in your mind, that how, from where Cl minus ion is coming, so Cl minus ion is coming from KCl solution. Okay, this KCl will be dissociated in K plus and Cl minus ion. Okay, I think all concepts are clear about this this oxygen sensor. Okay, chlorine oxygen sensor. Okay, and finally it will sense oxygen. Okay, so how much current is flowing? Okay, on the basis of that you can say that how much oxygen is there. Okay, if oxygen is more, then more reduction will be there. Okay, so the current will be more. Okay, and on the basis of that you can say how much oxygen is there in your analyzed sample. Okay, so this is the working principle and structure of chlorine oxygen cell. Is it clear? Is it clear or in doubt in chlorine oxygen sensor? Okay. Yes, yes. It is supporting electrolyte, which will means it is also oxidized. Means silver. Okay. If silver will be oxidized, so it need to balance, na? So it will react with Ag plus. Okay. So AgCl salt will become. Okay. So this is if you see, one is a reduction reaction and one is oxidation reaction. Okay. So by that way, the complete cell will be formed. Okay. Finally, you can say a complete electrochemical cell will be there. Okay. Uh, So now we will easy to answer this question. Okay. So now we know about Clark oxygen sensor perfectly. Which among the following is not component of Clark oxygen sensor? Okay. So ring shaped silver anode, yeah, it is there. Okay, we discussed now like that. Then cathodic platinum disc, yeah, it is also like that. Buffer HCl solution, no, because we use KCl and oxygen permeable Teflon membrane. Okay, so this is also correct. Okay, so finally C is wrong answer, and it is I think you do not have any doubt in this question. Okay, so this is the explanation of this question. The cell consists of cathodic platinum disc working electrode. Okay, so this disc electrode is working electrode. Okay, where oxygen is reduced in water. Okay, by 4 electron plus 4 proton. Okay, then where we are, working electrode embedded in centrally located cylindrical insulator. Okay, so cylindrical insulator is there, so it will provide insulation between cathode and anode. Surrounding the lower end of this insulator is ring shaped silver anode. Okay, if this is the, you can say, cathode, then here it will be like insulator. Okay, then finally a ring shaped electrode like that. Okay, so this is the perfect understanding. Surrounding the lower end. Okay, so it is surrounding the lower end of this insulator. Okay, so this is anode. The tubular insulator, this is tubular insulator. So tubular insulator and electrodes are mounted inside the second cylinder that consists of buffer solution of potassium KCl, potassium chloride or KCl. Okay, so it will be like that. So this solution will be KCl. Okay, a thin L20 mm repressible oxygen permeable membrane of Teflon or polyethylene is held in place at the bottom. Okay, so at the bottom a semi-permeable membrane of Teflon or polyethylene will be there. It is replaceable because you need to change it. Okay, frequently. The thickness of the electrolyte solution between the cathode and the membrane is approximately 10 micrometer. Okay, so this is this thickness. It is talking about this thickness. Okay, so this thickness is around 10 micrometer. So question number second is, so if you are able to answer question number first, then question number second is also similar. The working electrode of Clark oxygen sensor is, okay, same means nothing to explain here. Okay, cathodic platinum disc only working electrode. Okay, only two electrodes are there. One is a ring shaped silver electrode. Okay, which is anode. It is not used. It is also not. Okay, so these are two electrodes, but working electrode is cathodic platinum disc. Okay, so this is the right answer. The cell of Clark oxygen sensor consists of cathodic platinum disc working electrode embedded in centrally located cylinder insulator. Surrounding the lower end of this insulator is ring shaped silver anode. Okay, so it is very well explained. Now second sensor, it is about glucose sensor. Okay, so what is question? First we will read question. In case electrochemical glucose sensor, okay, electrochemical glucose sensor, the following catalyst reaction occur in glucose plus oxygen, glucose oxidase, which is enzyme. Okay, so its name says glucose oxidase means what is the work of this enzyme is it will oxidize the glucose. Okay, that's why glucose oxidase. Okay, so if you know a little bit about the biology, so its name say means oxygenase. Okay, means if some enzyme is generating oxygen. Okay, oxidase means it is oxidizing something. Lipase, okay, means its name itself suggesting that what is the working of this enzyme. Okay, glycan. So glucose oxidase means it is oxidizing glucose. So finally, see gluconic acid. So what is the acid formula? And generally in glucose OH group is found. Okay, so you can say ROH to ROH. Okay, so oxygen here you can say if one atom is there, then here it is two atom. Okay, so oxygen atom is attaching, so it is oxidizing. Okay, so glucose is oxidizing in gluconic acid. Okay, acid formula you know na, COH. This is the functional group, acetic acid you can say. Okay, if any acid is there, organic acid, I am talking about the organic acid, then there should be COH group. Okay, so finally glucose will convert in gluconic acid and it will also form peroxide. Okay, H2O. So obviously glucose is oxidized, so oxygen will be reduced. Okay, if one reactant is oxidized, then another reactant will be reduced. Okay, so oxygen is reduced. If you see here it is 0, oxidation state is 0, but here it is -2. Okay, okay, so this is all about this reaction. Then how this glucose sensor is made and what is the working principle, we will discuss a little bit. Okay, then we will answer. So glucose sensor is made of 3 layers. Okay, it is made of 3 layers. Okay, so first layer is made of polycarbonate. Okay, second layer is made of some you can say enzyme, and innermost layer is made of means you can say the layer which permit only very minute element, and it is generally made of cellulose acetate. Okay, okay, so these are the 3 layers. Okay, and what you are going to analyze? Glucose. Okay, so glucose is in your solution. Just a minute. Okay, just okay. So now glucose is in your solution. This is your suppose your test okay, and this is your test sample. So glucose will come in contact of polycarbonate. So this polycarbonate layer will permit only glucose. Okay, it is permeable to glucose only. So glucose will able to pass inside. Okay, and it will contact with enzyme. Okay, this enzyme is means little bit you can say jelly form or solid form. So it is immobilized, means it will not mobilize. Otherwise, if it is liquid form, then it will be pass, and it will, your sensor will not work properly. Okay, so it is in immobilized form. So finally glucose will come in contact with this enzyme. So what this enzyme does is, this enzyme is nothing but, you can say, glucose oxidase. Okay, this is the enzyme. Okay, so glucose oxidase, we can say GOX. Okay, glucose oxidase will convert glucose in gluconic acid here. Okay, in this layer. Okay, and so gluconic acid, we do not have any interest in gluconic acid. Okay, we are not going to measure how much gluconic acid is formed. We are going to measure how much H2O2 is formed. Okay, so in this layer H2O2 will be formed. Okay, plus gluconic acid will be formed. Okay, so we are ignoring this gluconic acid, only H2O2 we are worried about. So now on the innermost layer is made of cellulose acetate, and it will only permit to enter H2O2. Okay, hydrogen peroxide only will pass inside this layer. Okay. So here now only at the this is last layer, cellulose acid, which will permit only H2O2 to pass. Okay. Finally, this H2O2 will means which means contact with your electrode, and your electrode will sense how much H2O2 is coming, and on the basis of H2O2 it will give reading that how much glucose is there. Okay, so this whole calibration is done initially, and finally by this whole process, okay, you are refining your analyte. Initially glucose is coming, then glucose is oxidized in gluconic acid, and it will produce peroxide, and finally peroxide able to pass cellulose acetate layer and finally reach to electrode. Okay, so at electrode will sense this H2O2. Okay, how it will sense? It will react with OH minus ion here. Okay, and produce O2 H2O. Okay, so you can say suppose 2 H2O here. Okay, so and 2 electron. Okay, so finally you will get 2 electron. Okay, so again due to this electron, current will be flow. Okay, and this current will be responsible for how much glucose is there. If glucose is more, then more H2O2 will be produced. This reaction will be occur, the rate of this reaction will be more, and finally current will be more. Okay, current we will measure in ampere, so current will be more, and on the basis of this current you can measure how much glucose is there in your sample. Okay, so this is all about this glucose sensor. Is it clear? Glucose sensor is clear? Any doubt? Okay. Okay, so finally we will answer next question. Okay, let me erase. So this reaction occurred middle layer. Okay, where you can say immobilized enzymatic layer, middle layer. Okay, so this is all about explanation. I have already explained, but I will just read it out. The electrochemical glucose sensor that is widely used in clinical laboratory. Okay, clinical laboratory means generally it is used to measure oxygen in blood. Okay, for the routine determination of glucose in blood serum. Okay, so oxygen sensor and glucose sensor both are very useful in your clinical laboratory. Okay, because the if any person is going to detect any disease, okay, these are the main test for the diagnosis of any disease. Okay, so this is very common and very frequently used sensor in biological or clinical laboratories. Okay, this device is similar in construction of oxygen sensor. Okay, in oxygen sensor, or here we can say means overall it is same. Okay, means what is the working principle in that, how much current is producing. Okay, in the previous case we discussed that how if oxygen is coming inside, okay, then how much due to due to reduction of oxygen, how much oxygen is producing in oxygen sensor. Similarly, in glucose sensor, how much H2O2 is coming, and due to H2O2 how much electrons are generating. Okay, so it is also measuring current only. So in both cases, either it is oxygen sensor or glucose sensor, we are measuring current. Okay, the membrane in this case is more complex and consists of 3 layers. The outer layer is polycarbonate film that is permeable to glucose. Okay, it will permeable to glucose, but it will restrict another constituents like proteins and all whatever. Okay, so it will only permit glucose, but impermeable to protein and other constituents of blood. The middle layer is immobilized enzyme. Okay, so one enzyme we have already discussed, which is glucose oxidase. Okay, glucose oxidase in this example. The inner layer is cellulose acetate. Okay, finally, so it will permit only H2O2, cellulose acetate, which is permeable to small molecules such as hydrogen peroxide. When this device is immersed in glucose containing solution, which is your sample. Okay, glucose containing solution is your sample. Glucose diffuse. Okay, diffuse means, diffuse you know na, C1 to C2, concentration 1 to concentration 2, lower concentration to no, higher concentration to lower concentration. Okay, it is diffusion. So glucose will try to diffuse from higher concentration to lower concentration. So higher concentration will be in your sample. Okay, and lower concentration will be your sensor, glucose sensor. Okay, that's why glucose will move from sample to sensor. Okay, through the outer membrane into immobilized enzyme, that is middle layer, and glucose is oxidized in gluconic acid in this layer, plus it will produce hydrogen peroxide. Okay, so this is all about explanation and answer of this question. Just let me check. Okay, now question number 4. Proton motive force generated due to. Okay, see, proton, what is proton? Hydrogen plus motive force. Okay, so who can move proton? Simply in simple language we can say, who can move proton? Okay, so it can be moved. Okay, in two cases. First is due to diffusion. Okay, first is due to diffusion, second is due to negative ion. Okay, negative charge. Diffusion means if concentration is high here, so it will try to move in lower concentration. Okay, so first is this force, which is known as diffusion. Second is if you apply negative charge here, negative electrode, Okay, suppose this electrode is negative, so this hydrogen ion will attract toward negative electrode. We all know about that because it is positive charge, so it will attract toward negative charge. Okay, so these are the two forces. Okay, first is concentration and second is charge. Okay, so if you understand this thing, then you will able to answer. Okay, this question very nicely. Potential difference across the membrane. Potential difference means what? So this is your membrane here plus, okay, it is outside, and here suppose you applied one this is electrode. Okay, you are applied negative charge here, so due to potential difference this will be try to move inside. Okay, so potential difference across the membrane, yes, it is right. Second, difference in proton concentration across the membrane. What is this? This is nothing but diffusion, because difference in proton concentration. Okay, see, suppose okay, here H plus has C1 concentration and here hydrogen plus has C2 concentration. If H plus C2 is lower than HC1, okay, so what will happen? So this is the bigger concentration. Okay, so hydrogen ion will move inside. Okay, and it will move inside due to what? Due to concentration. Okay, or diffusion. So these both cases are explained here. Okay, so I think you can answer this question very nicely now. Understood this question and answer? Okay. Okay, so this is all about question 4. And proton motive force is the force that promotes movement of proton across the membrane. Downhill, the electrochemical potential. Okay, proton motive force is generated due to potential difference across the membrane and difference in proton concentration across the membrane. Okay, so these both two factors are responsible for proton motive force, or you can say proton movement force. Okay, how proton will be moved. If you know about the proton, if you know about the charge and whatever how it will be moved, then you will be able to answer. Okay, so here two things are means you need to know about electrochemistry as well as what, simple chemistry. Okay, where diffusion and all things are taught. Okay. Sorry. Okay, then question number 5. Among the following, the incorrect statement about the electrochemical glucose sensor is. Okay, it is already discussed, glucose sensor. So it consists of 3 layers, right? The inner layer is permeable to H2O2. Yes, it is made of glucose acetate and it is permeable to small molecule like hydrogen peroxide. So this is also correct. The middle layer consists of immobilized enzyme. Yes, it is made of glucose oxidase. Okay, which reduce glucose in hydrogen peroxide and gluconic acid. Okay, so this is also correct. Finally, the outer layer is permeable to both glucose and protein. No, okay, it is a barrier for protein. Okay, it is a barrier for protein. It will only allow to pass glucose. That's why this is wrong answer. Okay. So outer layer is only permeable to glucose. Inner membranes allow phases of hydrogen peroxide, which is then oxidized to oxygen emitting 2 electron. Okay, whatever this reaction is occur where it is occur at electrode. You can say innermost, after innermost layer electrode is there, where this reaction will be happen, where it will produce 2 electrons, which is responsible for current flow. Okay. Then question number 6. the gas produced in microbial fuel cell and microbial electrolysis cell are. Okay. So microbial, what is a microbial fuel and what is microbial electrolysis? Okay. So this microbial word is common in both things. Okay. So means some microbes are doing something. Okay. Means here microbes are using to use as a fuel cell, where here microbes are used as a electrolysis. Okay. So how it will be happen? Okay. If you will discuss about that, then you will be able to answer this question very nicely. Okay. So I will draw here, but it is not. I will not able to draw perfectly, but I will try to explain. Okay. Suppose there is so first of all we will say about microbial fuel. So any load, suppose you are providing one bulb here. Okay. So this bulb is glowing, means this cell is generating some energy. Okay. So this is the you know why we are using cell. Okay. We use cell. Okay. Due to get some electric energy. So this cell is also generating electricity, but how? Okay. So we can say, suppose from here waste water is coming inside. Okay, waste water means it will contain many impurities. Okay, and this is microbial cells, so some microbes will be here. Okay. So what these microbes will be do? Okay, it will oxidize. It will oxidize whatever impurities are there in your waste water, organic, okay, inorganic, whatever. So it will oxidize. So it will be oxidized. So whenever oxidation is there, okay, it will produce CO2. Okay. So it you can say here, whenever oxidation is there, okay, something is oxidized, so electron will be produced. Okay. If something is oxidized, the electron will be produced. So electron will be flow like that. Okay. And CO2 will be formed at here. Then this electron will come here. Okay. After means this is the circuit. Okay. So finally it will be here. And from here, in this air, you are providing air. Okay. Air means oxygen. So what will happen? This electron which are coming here will react with oxygen. Okay. And it will reduce oxygen. Okay. It will reduce oxygen. Minus, we can say, suppose like that. Okay. So this is all over your means working principle of this thing. Okay. It will not NO that noise cathode. Okay, this is all about this. And finally, from here, effluent will be removed. So what is effluent? Initially you are Initially you are providing wastewater in your fuel cell. So all the we cannot say all, but the impurities which are present in wastewater will be oxidized by microbes, and it will form CO2 plus electron. So this electron will provide in at anode, and due to this electron, the electricity will be flow, and it will create some voltage, and you can use this voltage as your external source. Okay, external, you can say, you can consume this potential. Okay, so it will be work as a electro biased fuse, and finally this effluent will be recovered. Okay, after most of the impurities which are present in wastewater will be oxidized. Okay, so this effluent will be means you can say you use this wastewater to produce electricity. Okay, so it is very good for sustain energy. Okay, so this is all about how your microbial fuel cells work. Okay, is it clear how it is working? Okay, then second thing is how microbial electrolysis is there. Okay, how microbial electrolysis is there. Okay, so we need to erase it again, but we will not erase all things. Okay, now we will discuss about how electrolysis will work. Okay, see now electrolysis. Okay, so in fuel cell always generate electricity, but in electrolysis we need to provide voltage from outer source. Okay, so here we need to apply one battery. Okay, so big vertical line always representing positive electrode, and small vertical line represent negative electrode. So this is your battery, and you are providing voltage. Okay, these are two electrodes. So here little bit difference. So from here hydrogen will be generated, and from here CO2 will be generated. Okay, so how it is there? Okay, so finally this is the negative terminal, so electron will be flow in like that. Okay, here electron will be there, but this is the positive electrode, so electron will be flow of electron will be like that. Okay, so electron flow will be like that from this direction to this direction. Okay, first of all this thing is clear. Now how it is happening? So microbes will help to reduce impurities from wastewater. Similarly here also. Okay. So here it is electron. Okay, so suppose H plus ion is there. Okay, it will gain electron, can produce hydrogen gas, which is here. Okay, so you will get hydrogen gas here. Okay, similarly, because here it is reducing, it is reduced. Okay, so here oxidation will be there, okay, and it will produce CO2. So CO2 will be removed from here, okay, and similarly effluent will be released. So these microbes are helping in electrolysis, okay, and finally by this way you are able to produce hydrogen gas also, which can use for fuel purpose, okay. So this hydrogen ion and all these things are generated by microbes, okay, and by this way you can do electrolysis. Okay. So this is the working principle of microbial fuel cells. So the gas produced in microbial fuel cells and microbial electrolysis cell are, okay. So see here hydrogen is producing, okay. CO2 is also produced, but CO2 is not produced. Sorry, hydrogen is not produced in your electrobial cell. Okay, so right answer will be CO2 in this thing, and here both gases are producing, but hydrogen is producing. Okay, so this is the right answer. CO2 and hydrogen respectively. Okay, so B is right answer. So this is the explanation of this question. Microbial electrolysis cell (MEC) is part of the microbial electrolysis cell technology, which is one of the renewable energy. Okay, renewable energy means energy which can be replenished. Okay, so this we are replenishing our energy source. Now we are using wastewater. Okay, so from wastewater we are generating energy and gases like hydrogen, which can be used as a fuel. That's why it is very this is the source of renewable energy. Okay, renewable energy alternative today. MEC, microbial electrolysis cell operating, is based on fundamental of bioelectrochemical process and is a promising renewable energy technology that produce hydrogen gas. Okay, however microbial cells CO2 is formed. Okay, so it is forming CO2, but also it is giving you electrical energy. Okay, here it is not, here you are providing voltage or you are providing electrical energy and it is producing hydrogen. Okay, so these are the cases of this. This is all about MEC, microbial electrolysis cell, and microbial fuel cells. Is it clear? Any doubt up to here? Yes, MEC both gases are produced. Okay, but according to answer we can say that means CO2s are producing in both cases, but hydrogen is producing only MEC. That's why we are. Okay. Question number 7. In metallurgical microbial fuel cells, metal is recovered. Okay, see what is happening here. This concept we have already discussed. Okay, metal is recovered at what? Okay, so metal generally metal is formed in we can say M plus form. Okay, so you need to reduce it and it will form metal, oxidation state 0. So it will be solid metal. Okay, we discussed this example now. Suppose it is copper Cu plus 2. Okay, when it will gain 2 electron, it will make copper metal. Okay, so this reaction will happen at where? Okay, at negative terminal, because it need 2 electron. Na? Okay, so suppose these are the your cell. Okay, so you can say this is the negative electron, this is the positive electron. Okay, so here at negative electron this reaction will be up. Okay, where your any metallurgical means we are discussing about the metals. Okay, so metals will gain 2 electron and produce metal, and this reaction will be occur at negative electron, which is known as a cathode. Okay, that's why cathode is gaining weight and all things we have already discussed in detail. Okay, so cathode is right answer. Okay, understood? I think this question is already taught in previous sessions. Okay, clear? Question number 7. Okay, so metallurgical microbial fuel cells is a attractive alternative for recovery of copper. Okay, so why we are using this type of microbial fuel cells where concentration is very low. Okay, generally in industrial and industrial use and for laboratory use also we generally use different techniques, but if your copper concentration is very low and it is in wastewater, okay, not only copper, any metal, okay, if concentration is very low and it is in wastewater, so you can remove it from wastewater. Okay, that's why it is very attractive technology. Okay, so for recovery of copper from copper containing waste stream, okay, waste stream, as the metal is recovered in its metallic form at the cathode, while the energy from the metal reduction can be obtained from the oxidation of organic metal at anode. Okay, so oxidation will be occur at anode. Okay, and reduction, copper is gaining electron, na? So it is reduction, so reduction will be occur at cathode and oxidation occur at anode. Okay, so this is done. Then question number 8. The enzyme catechol oxidase the enzyme catechol oxidase catalyzes the conversion of. Okay, so first of all you just I think you know about this structure, but just this is the phenol. Okay, and if it has two hydroxyl group like that, okay, in ortho position. Okay, this position is known as a ortho position, just adjacent of one hydroxyl group, so it is known as a ortho diphenol, or you can say it is a catechol. Okay, this is the general name of this compound. This is catechol. Okay, this is the catechol. This is the phenol. Okay, this one is phenol. So the enzyme catechol oxidase catalyze the conversion of. So this is catechol, and if you use catechol oxidase, means what? Oxidase means it is doing some oxidation. Okay, its name says means it is doing some oxidase. Okay, so when catechol will be oxidized, okay, this 2H will be removed. Okay, so when this 2H will be removed, it will form compound like that. Okay, now you need to satisfy carbon valency remaining. Okay, so this type of compound will be formed. Okay, so here see what happened, 2 hydrogen is removed. Okay, so you can write here 2H plus is removed. Okay, and how much electrons are gained? So you can say 2 electrons are gained. Okay, so finally this is the oxidation. Okay, this reaction is oxidation because you are using catechol oxidation. So finally oxidation is occurring of catechol to this compound is known as phenol, orthophenol, because oxygen groups are in ortho position, so it is known as orthophenol. Okay, so this is all about this reaction. Okay, so now you can answer this question. So this catechol is also known as the orthodiphenol. I have already written here orthodiphenol. Okay, 2 phenolic group is here, that's why it is diphenol. Okay, and it is ortho position, so orthodiphenol position to orthophenol. Okay, so from here to here this reaction is occurring due to catechol oxidase. Okay, so that's why the B answer is right. Okay, clear? I think this question is clear. Okay, so catechol oxidase is a copper oxidase that contain type 3 dicopper cofactor. Okay, so 2 copper are there, dicopper cofactor, and catalyze the oxidation of orthodiphenol into orthoquinone coupled with reduction of molecule oxygen to water. Okay, it will also oxygen, and we gain 2 electron also. If you remember, then we gain 2 electron. Okay, so it will reduce oxygen -2. Okay, like that. So 2 electron more means it, you can say only half of it. Okay, so like that. So finally it will be there. This reaction will be also there due to 2 electron produced in our previous case. Okay, I think you know how it is produced. It is produced due to catechol to quinone. Okay, orthocatechol to orthoquinone. When it is produced, it will make 2 electron. Okay, so this electron will react with oxygen and produce water. Okay, is O minus nothing. It will react with 2 proton. Okay, and finally it will make 4 proton. So here you need to, if you want to balance it, this reaction, just a minute, I need to balance it because it is not 2 oxygen. Okay, 1 oxygen only. Because I write only half O2, so half O2 means 1 oxygen, reduced. Okay, then there is 4 electrons, so it will make H2. Okay, so this is all about how catechol oxidized in orthoquinone in presence of your what say catechol oxidase. Okay. Finally, question number 9. Which among the following is not a polyphenol based biomolecule? Okay, this is the memory based question because structure of this compound is very typical. Okay, so you can say it is xylose, because xylose generally we know that sugar, xylene sugar we use. Okay, so xylose is similarly, but another these all compound containing you can say phenol, not phenol, what say amide group. Okay, that's why it is different. Okay, so see, lignin, tannin, and flavonoids are polyphenol. Okay, it is contain polyphenol, a large family of naturally occurring organic compound categorized by multiple of phenol units. Okay, it is contain phenol only. Okay, best biomolecule where xylose, a kind of pentose sugar. Okay, it is a pentose sugar. Pentose means 5 carbon. Okay, so it is kind of pentose sugar. Its molecule formula is this one. Okay, so no need to, if you have interest then you can go, but otherwise no need to understand what is the formula. It is a very complex thing and it is also not part of this course. Okay. Finally, question number 10. The enzyme used in active layer of polyphenolic bisensor is R. Okay. See, now the enzyme used in the active layer of polyphenolic biosensor. Okay. So phenolic biosensor. Okay. So how it is work? So we will discuss about little bit. Okay. The answer is very simple, but I will explain you how it will work. Okay, so phenol, as we discussed in last or second last question. Okay, this is the oxygen. This is the structure of phenol, where oxygen has 2 lone pair. I think you know why oxygen has 2 lone pair. Okay, lone pair means what? This is electron rich site. Okay, I will give you little bit about this insights. It will be very helpful to understand this mechanism. Okay, if you see the structure, okay, it is the electronic structure of oxygen. Okay, 1s2, 2s2, 2p4. Here, if you fill the electron in outermost shell, this is the 2s. So both electrons are filled. It is 2p. So there are 3 orbital. Okay, now we will fill electron. So 4 electron we need to fill like that. Okay, so this outermost shell will take part in chemical bonding. So it will be hybridized and it will make sp3 bonding, sp3 hybridization. Okay, so 4 orbital will be there. You can say 4 orbital will be there like that. All are sp3. Okay, out of that 2 orbital has already fully filled. Okay, so these fully filled orbitals are represented by lone pair. Okay, so you can say in very good thing. Okay, by dot it might be little confusing, but we can say we can draw this because these are the orbital, na? So we can draw complete orbital, and this 2 orbital has this 2 electron like that. Okay, so these are known as a lone pair. Okay, these 2 things are known as a lone pair. Here, this is nothing but this fully filled orbital. Okay, now these are the 2 means you can say half filled orbital. So it will make bonding. Okay, so it you can say oxygen can make 2 bonds. So here oxygen has make 1 bond with hydrogen and 1 bond with this carbon. Okay, so finally this 2 lone pair is there. So oxygen is electron rich. Okay, so copper ion, suppose we are taking any metal ion, so I am taking copper here. So copper can attract by this lone pair because it is positively charged. So it will be attracted due to this negative lone pair, and when it will attract this side, okay, electron will copper will pull this electron. Okay, so finally oxygen will feel that it is electron deficient because oxygen electrons are pulling by copper. So oxygen will feel deficiency of electron, so it will pull electron from this side. Okay, finally it will pull electron from this side. So this side will become your little bit electron deficient, or you can say active side. Okay, so it will this side will this side will called as active side. Okay, so and that's why this side will oxidize first. Okay, and finally if you see this phenol, when it will be oxidized due to this oxidase enzymes. Okay, catechol oxidase, whatever, not catechol oxidase, simply oxidase you can say. So it will oxidize here and it will make catechol like that. Okay, diphenol, from phenol to diphenol. Okay, so this is the mechanism of this reaction. Okay, so only due to from here to here only one oxygen is used. You can write half oxygen, and here the enzyme which is used is known as a monooxidase. Okay, why it is known as a monooxidase? Because it is consume only one oxygen. That's why it is known as a monooxidase. Okay, so now catechol is formed. Catechol form, or you can say orthodiphenol. Orthodiphenol is formed. Okay. Now further reaction we have already discussed. Okay. So if it is a catecholase, if you use, then what will happen? This compound will convert in orthoquinone like that. Okay, so this is all about your sensor, phenol sensor, how it is work. Okay. So the enzyme used in the active layer of polyphenolic bisensor is R. Okay. So both B and C, nitrogenase we have already discussed. Okay. And lacase is also used for this type of these enzymes. Okay. Because monocatagonous category, this is only one type of example. But if you read further, then different type of enzymes are being used. Okay. So these are the answer or explanation of this question. Biosensor for phenolic contact assessment. Okay, phenolic contact assessment. The biosensors for phenolic compound mainly incorporate two enzymes, tyrosine and lactase. Okay. So this is all about assignment 10. Okay. And we further discuss about some concepts. Okay. About this thing. So we will take 5 minutes break. Okay. Then we will again discuss some remaining topic. So please refresh and join after 5 minutes. Okay. Okay, we should start now. So here in this slide, I added this slide, okay, to explain you how oxidase enzyme and how oxygenase enzyme work. Okay. So if you see here, oxygen reacting with hydrogen plus 2 electron, it is making water. Okay, so it is reduction. Okay, so it is making oxygen. Then uh, this M, this is the metal ion, okay, which is oxidized, okay, which is oxidized due to this reaction and making Mn plus 2 oxygen. Okay, means it is making it is oxidized, it is further oxidized, N plus 2, N plus 2. Okay, then finally this is substance, and substance is converting in means this oxygen atom is attached now with this substance. Okay, okay. So this is the oxygenase, means oxygen is generating here. Finally, by this complete process, this substance is gaining oxygen. Okay, that's why it is known as a oxygenase process. Okay, oxidation plus it is gaining oxygen. Okay, this is H2B means substrate. Okay. So this is the oxygen atom transfer. Okay. So what is happening here? Oxygen atom is transferring. Oxygen atom is transferring from here. Okay, here to here, here to here. Finally, it is so because it is the oxygen transferring. Okay, that's why it is known as a oxygenase mode. Then what is the oxidase mode? Okay, because name is similar. Oxidase mode means it is responsible for oxidation, but it is not generating oxygen. Okay, how it will be if you see this reaction here also oxygen reacting with a proton and it is making water or hydrogen peroxide. So by this reaction it is oxidizing. Okay, it is Mn plus, it is oxidizing Mn plus 2. Okay, so it is oxidizing. This reaction is oxidization, but oxygen is not generating. Here oxygen is also generating. Okay, so if you see further, this substance which is attached with hydrogen, okay, it is lose hydrogen. Okay, it is lose hydrogen, so it is also oxidation, but it is oxidation is not attached with this substance. Okay, so oxygen is not generating, but also the substance is oxidizing. And how it is oxidizing? It is oxidizing due to removal of hydrogen or due to losses of hydrogen. That's why it is known as a oxidase mode. Okay, so this is all about. Okay, so this is all about this week assignment, and okay, so this is the means at the end we discussed how electrochemistry is useful for your analytical chemistry. Okay, electrochemistry means chemistry which is related to electricity. Okay, means by chemical reaction you are producing some signal which can be explained by electricity. Okay, means suppose you are producing electron, so this electron can be analyzed or can be measured by electricity, by current, by voltage, whatever. Okay, so by that way you can measure different type of constituents in your electrolytes or your sample. Okay, so these all are that's why it is very useful for your analytical chemistry. Okay, so in analytical chemistry from starting, where we used, you can say by flame you can detect some metal like sodium, potassium. Okay, similarly you can use volumetric analysis like titration. Okay, from where you can analyze different type of iron. Okay, different type of molecules. Okay, by titrations, iron atoms, elements you can measure by titration. Okay, then you can, if your concentration is very low in your sample, then you can use some very highly sophisticated instruments. Okay, and spectroscopy. like atomic absorption spectroscopy, atomic emission spectroscopy. Okay, nowadays ICP, and ICP is inductive coupled plasma. Okay, which is very advanced technique. So then you can measure a hydrogen ion, calcium ion, sodium ion. Okay, by some electrodes which already discussed previously. Okay, so there are different type of method. Okay, where you can use your chemical knowledge, and this chemical knowledge or chemistry knowledge you can club with another techniques. Okay, and so this type of techniques will finally useful for many areas. Okay, so you already know that it is explained by sir that suppose we discuss about the electrochemistry. Okay, so electrochemistry is used for analysis. Okay, it is analysis means suppose you are going to measure very minute concentration of copper ion, silver ion, gold ion. Okay, so you can use electrochemistry there also. Okay, in biological sample also, like sensors. Okay, we discussed already how glucose and oxygen can be analyzed. Okay, in environmental purpose also. Okay, that how much pollutant are there in your water, in your air. It can be it can be also analyzed by electrochemistry. Then in materials. Okay, materials, in material science you can use this type of electrochemistry, like okay, in how material science, because if you know the gold plating. Okay, so in older days all yes, actually I am explaining only, slides are not there, I am just explaining these things. Okay, so uh, material science, like in older days only gold plating are means only gold ornaments are there, but nowadays we can use only minute amount of gold, and we can plate. Okay, it is known as electroplating. Okay, so you can plate precious elements like gold, platinum. Okay, on the other elements. So, and it is not only for ornaments, some to use some scientific study also we plate some electrodes by different elements. Okay, so simple example is iron. Okay, iron is very you know that iron in presence of moisture and water it will be corrode. Okay, so it will be oxidized and it will make Fe2O3. Okay, and it is finally oxidized. So you are means you are losing iron because it is oxidized. Okay, and whatever structure you have, it will lose its mechanical strength. Okay, it's shiny, whatever. Okay, so it is a undesirable reaction, but it will be happen. So what we just use electroplating. And which element we use, anybody has knowledge which element we use to protect iron? Very good. Yes, we use zinc. Okay, so we use zinc. Okay, so what zinc will do? Zinc will first sacrifice itself. Okay, sacrifice itself means zinc will convert Zn plus 2 plus 2 electrons. Okay, so it will be oxidized. So because zinc is oxidizing, so iron will your moisture and oxygen whatever is there, it will not able to reach till iron. But if you see after some time, after some years or whatever, it is depend on it is depend how thick layer of zinc is there and whatever conditions. Okay, is it acidic condition and whatever. But finally after some time the iron the zinc layer will be removed, and after that iron will be corrode. Okay, iron will again start corrode. Okay, so this process is known as galvanizing. Okay, you are providing a layer of zinc outside the iron. Okay, this is the zinc layer and this is the iron. So this process is known as a galvanization. Okay, similarly sometimes we also use a sacrificing metal. Okay, suppose this is made of iron and it is made of magnesium. Okay, and you connect it with the iron, and suppose this is the iron pipe. Okay, this is the iron pipe which is responsible for water flowing. Okay, water supply, and you attached this this pipe with magnesium. Okay, so what will happen? magnesium will oxidize, okay, and it will produce 2 electrons. So these 2 electrons will be there, okay, it will be reached to the iron and it will be electron rich now, so it will protect from oxidation, okay. So this reaction will protect iron from corrosion, okay. So what is happening? So electron is coming from here, okay, so it is sacrifice, you can say sacrificial metal, okay. So magnesium is sacrificing itself to protect your iron, okay. So this type of technology also used in, so this is all about the electrochemistry, okay. So this use of this understanding of these concepts you can also use to analyze how much this type of elements are present in your analyte, okay. So this is about material science, how you can use electrochemistry in material. This is only electrochemistry, no? How means which element is sacrificing, and do you know means which element will be sacrificed first, and how you will decide that which element will be sacrificed, okay. So it is all depend on your what say electrochemical series, okay. So hydrogen we say that its electron reduction potential is 0, okay. So element above the hydrogen and electron below the hydrogen, okay. So suppose here, suppose I am writing lithium, okay. So lithium, what suppose it is lithium here, or no, not lithium, just a minute, let me write so it will be very clear. Suppose you are writing fluorine here, okay. Or we can say Ag, silver plus gain electron and converting in silver, here zinc losing electron and it is oxidized, okay. So it is depend on that, okay. Means if you want to protect silver, okay, then you can use zinc, okay, because zinc will be oxidized, okay. It is its a reduction potential, its reduction potential will be negative, okay. But because it is oxidized easily, so its oxidation potential will be positive and its reduction potential will be negative. Similarly, here its reduction potential will be positive and here oxidation potential will be negative, okay. So you can use this zinc to protect silver, okay. So it is all depend on electrochemical series, that which elements you can use to protect which element, okay. How which metal you want to save from the corrosion, okay. So it is also very useful in material science, okay. Then also in food, okay. So what we can say, just a minute, you can say in bioelectrochemistry, okay, in biological, how what in a, okay, in biology, first of all it is in biology, then electrochemistry. So the reactions which are occurring due to electron transfer, okay. In biology, electrons reactions which are occurring due to electron transfer is studied in bioelectrochemistry, okay. So simplest example is, suppose it is food, okay. So food, whatever, glucose and whatever, so when it is oxidized, okay, so it will make CO2 plus water plus one more important thing it will produce, which is energy, okay. So this energy is required for metabolism and other activity for our body or other living being, okay. So here what will happen, this because it is you can say oxidized, okay, it is oxidized, so it will give electron, okay. and this electron will be responsible to convert ADP, which is adenosine diphosphate, to adenosine triphosphate, like that. Okay, so this process is done by electrons which are coming due to oxidation of food. Okay, so this is, you can say, bioelectrochemistry. Okay, it is also very useful, and here also some enzymes which enzymes play important role. Okay, and if any enzymes contain metal ion, okay, then it is known as a metal enzyme. Okay, and which is used for electron transfer reaction. So electrochemistry is also used as biosensors, biofuel cells, bioelectrosynthesis. Okay, and as we have already discussed about the microbial fuel cells and microbial electrochemistry, so I will not discuss it again. Okay. Some enzymes also very useful to recycle nitrogen in our system. Okay, so this is urea, which is produced due to metabolism of our body and other organisms. Okay, so in the presence of urease enzyme, urease means it will you can say consume urea. Okay, and it will make ammonia plus CO2. Okay, so this type of enzymes also used to produce ammonia and CO2. Okay, then this ammonia can be consumed again, and it will be source of your nitrogen for soils and okay. So this is all. Okay, so finally we have done this course, analytical chemistry. Okay, we have discussed what are the techniques. Okay, like you can say from the volumetric, okay, flavometry to volumetric analysis to spectroscopy, from spectroscopy to electrochemical technique. Then we also discussed about the thermo techniques. Okay, TGA, gravimetric analysis, DSC, okay, PGA. So different techniques we have already discussed. Okay, so these all techniques are used to analyze different type of analyte. Okay, and it is depend on what is the form of your sample, what element you are going to use, what compound you are going to use. Do you want to measure weight? Okay, how much weight is gaining or losing, then we will use TGA, like that. Okay, if you want to analyze phase transition, then also TGA. If you want to analyze elements, okay, then elements also depend on that whether you want to analyze qualitatively or quantitatively. Okay, if you want to analyze qualitatively, then different process. If you want to analyze quantitatively, then you need to more sophisticated. Okay, because quantitatively means you need to aware in each and every step. Okay, there should be not sample loss. Okay, in each and every step of your analysis, otherwise you will not get correct reading. Okay, so from older days where we use volumetric and gravimetric analysis, nowadays many sophisticated instruments are there where you can read in ppb level. Okay, ppb means parts per billion. Okay, so it is very minute level. So if your water contain cadmium, lead, cobalt, this type of hazardous elements. Okay, but the concentration of these elements are very low, so we cannot go with these techniques like titration and flame photometry. Okay, because concentration is very low. Okay, so that's why you need very sophisticated technique. Okay, like ICP and ICP, and further you can say electrochemistry techniques. Okay, electrochemistry sensors. Okay, which can read and which can analyze your sample in ppb level. Okay, so this is all about I think you people have make an idea how chemistry and other field, electrical field, you can say, knowledge, and also you can say thermal knowledge. Okay, in your in analytical purpose. Okay, so you can analyze your sample perfectly. Okay, So I am going to end here only. I hope if one more session will be there, which will be summarize session, okay, where we will discuss some, where I will discuss all the concept which is taught in this session, okay, in this course, okay. Till then I am stopping here only. If you have any doubt, you can ask. Otherwise, I am going to stop here only. Okay. Yes. Okay. 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 Then best of luck for your exam. Prepare well. Okay. 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 Bye. Okay. Thank you.